Alright gang, here we are with our video for Chapter 18, Section 1, and this one is specifically focusing on viruses. Lots of terminology in this chapter, so you kind of have to just make sure you learn what the terms are, and I'm going to try my best to explain what they mean as we go through the series of videos here. So, first of all, viruses, if we just take a look at what they are, they call them infectious particles. They are not actually living organisms or anything like that, but they can cause infections, so they call them infectious particles, and those particles will always be made up of, one is a capsid. Capsid is just another term for proteins, a series of proteins. That capsid will be surrounding some form of a nucleic acid, so that could be DNA, either double-stranded or single-stranded DNA in there, or it could be RNA, either double-stranded or single-stranded RNA. Could be inside of a virus, so we have a nucleic acid surrounded by a protein, and on some viruses we have something we call an envelope. So here's a bunch of pictures of viruses from the books, and if we just kind of zoom in on some of these, it would be nice if, let's move these out of the way. The first one's here, just terminology-wise, the capsid, if you remember, is the protein coat that's around the nucleic acid. So here we've got one type of virus. You're seeing the protein coat is this purple-colored things here. That's the capsid, made of capsomeres. You'll see that number, or that term spoken of a lot. Capsomeres are basically the individual building blocks of the capsid, or the protein coat. And then inside of both of these, this has RNA, so that's a nucleic acid. It's coiling down through the middle of this. That's the tobacco mosaic virus, which it talks about a bunch. And then this one right here has DNA in the middle, and that would be the virus for the common cold. So that's those. Some other things just to point out on other ones. This picture here is of something called a bacteriophage. Oopsies, bacteriophage. And the book talks about that a ton. It's just another type of virus, but they specifically target bacteria, and the term phage is often used for these. And again, it's just a very complex protein coat in this case, or capsid around here with these tail fibers and just a bunch of different things. But then the DNA is held in the head of this bacteriophage. These have been studied a ton. That's why they get pointed out specifically. And then the last one here, what we're seeing as different is our optional piece, the envelope. Okay, and the envelope basically, if I look inside of this, we've got our capsid, or protein, covering our RNA, which is our nucleic acid, which is noticed in the red there. Okay, and then we've got a couple of these surrounded by a membrane, and that's what the envelope is. This membrane would have come from whatever the host cell was that this was originally a part of when this virus was made. This would have come from the membrane of the host cell would have surrounded this, and then we've got glycoproteins, which would have come from the host as well. On the outside of this one, we'll talk a little bit later about some viruses that specifically have membranes and how they work out. Okay, so that's a basic there of what's involved with the virus. You just have a nucleic acid surrounded by a protein, and every now and then they've got a membrane around that we call an envelope. So that's that. Now, the rest of the chapter is basically dedicated, or this section, sorry, the rest of the section is dedicated to reproduction within viruses. So let's go ahead and take a look at the basic scheme of reproduction for a virus. And here we have it. This would be the basic overview of reproduction for a virus. There's a bunch of what we will find out is every single virus has a different mechanism for exactly how it goes through all the steps of making more of itself. But one thing they all have in common is these viruses need to have a host cell. So we have this host cell down here. Viruses can't reproduce on their own, but once they're in a host, they can basically make the host a virus-making machine, is what they'll do. So they'll turn this host cell into a virus-making machine. So the general plan is somehow the viral DNA has to get into the host. Okay. Sometimes the complete virus goes into the host, and then the DNA comes out of the virus. Other times the capsid stays on the outside, and the DNA, or whatever the nucleic acid is, will enter the host. Once that nucleic acid is inside the host, it's basically going to take over the host. Most of the time it will oftentimes it'll destroy the host DNA, and then it'll use the host's machinery to make copies of its own DNA. Okay, so we're making a bunch of copies of its own DNA, and then also to do transcription and translation to make all of its proteins, those capsomeres, the little building blocks. It's going to make a bunch of those. So we have entered the cell, we have copied or replicated our DNA, we have done transcription and translation to make our proteins, and then we're going to assemble all of that into a new virus. Assemble all that into a new virus. Once we've assembled new viruses, somehow those assembled viruses get released. Okay, so again, we enter the cell, we make new nucleic acids, we make new proteins, we assemble them into new viruses, and then somehow we release the viruses. So we're going to get to some more specifics on this, but that was the general overview of a virus replication cycle. So we're going to look at the lytic cycle here really quickly. Lytic cycle, and this is specifically looking at the bacteriophage. These are the most commonly studied bacteriophage, which, if you remember, they specifically attack bacteria and are very easy to study, which is why they're very well known. So this lytic cycle, first of all, if we have a cell or a virus that only reproduces by the lytic cycle, that's called a virulent phage or a virulent phage. Also note, this lytic cycle, I've also heard it called the lytic cycle. But anyway, this basically mimics what I did on the previous slide. Just some specifics to it are included. So first of all, we have 5 steps, and they are numbered here 1 through 5. So number 1, we have the attachment of the virus, and so there's going to be some kind of recognition that's happening between the virus and the bacterial cell. So there will be receptor proteins typically on the surface of the bacterial cell, and those receptor proteins are recognized by the proteins on the capsid of the virus. Okay, so there's this attachment happening. Once attachment has happened, then we're going to have the DNA, the nucleic acid, enter the host. Okay, and in this case, you're going to notice with this T4 phage, you're going to notice that the capsid stays on the outside while the DNA is injected into the cell. Once the DNA goes into the host cell, it's actually going to facilitate the breaking down of the host DNA. So here you can see the host DNA is breaking apart. Once we have that entry, then the bacterial DNA is taking over this cell and using it as a virus-producing machine. So we are going to be making all kinds of copies of the DNA of the virus as well as of the proteins of the virus. So this is the synthesis stage. Once synthesis is done, then we're going to have the assembly stage. Okay, where we are just going to assemble new viruses. So we have all the pieces together. Now we're going to put them together, and you can kind of see this broken down over here. But put them together, and then we've got a bacterial cell that is full. It's loaded with viruses, and those viruses are going to break out. So it's the release stage here, release. And they say that the cell is lysing, and so you'll have a bacterial cell that was infected with one virus, and then once it lyses, it's going to release 100 or 200 virus particles, and so you can see you can go from one virus infecting a bunch of bacteria to 100 viruses after one cycle. It would be 10,000 viruses after the second cycle. Once all of these leave and go into their own new host cells, you'd end up with 10,000. The next round, if all of those infect more bacteria, all of a sudden you get up to 1 million viruses. And so you can see this can happen very quickly, that you could wipe out a complete community of viruses. So, a community of bacteria with these viruses. So that's the lytic cycle. Now we're going to look at a slight variation of the lytic cycle called the lysogenic cycle. This is the diagram for the lysogenic cycle of a lambdaphage. It's another type of bacteriophage, but this one, typically cells that do the lysogenic cycle, they will also do the lytic or the lytic cycle. Those type of phages are called temperate phages, if they can do both. And so the scenario, if it went through the lytic cycle, it would do the exact same thing as the previous slide, and that's what's shown over here. But there's another option. Once the lambdaphage attaches and then has the entry of its DNA circularizes so that it is similar to a bacterial DNA. If you remember, bacterial DNA is in a circle. So it makes a circle. If conditions are correct, okay, at this point it will actually take over the cell and go through the lytic cycle. Okay. But if conditions aren't correct for this to take over the cell, and we'll talk about that in a second, what it'll do instead is it'll take its own DNA, the virus will, and it'll insert it into the middle of the host DNA. Okay, the way it does that is it's kind of similar to crossing over when we learned about that back with meiosis, with the crossing over of DNA. It's a similar thing where these get very close to each other, and then there's specific locations where they will enter into the host DNA. At this point, this is going to be called a prophage. Okay, this DNA, the viral DNA that's inserted into the host DNA, we call this time period a prophage. During this time, basically the virus DNA is going to be silent for the most part. The host will be doing everything that it normally does, but there's just this extra DNA that it's carrying. Okay, there is a chance. There's a couple cases where the viral DNA, as the cell's going through its life, some of the viral DNA will be used to make certain proteins that can change phenotypes of the bacteria. But for the most part, oftentimes it is a silent situation for the virus. Virus is dormant. Then that virus is going to, that viral DNA, the prophage, is actually, as this cell goes through replication, okay, and we make two new cells, so this is showing the copying of the DNA replication, and then this would go through binary fission to break the cell in half, and we'd have two new bacterial cells. Both of those bacterial cells will have the viral DNA because it got replicated also. Okay, so this will split off, and we'd have another cell here where there was a prophage in it, and that can happen over and over and over and over again. So you end up with a whole lot of bacteria that all have the viral DNA in them. Okay, so we have this prophage. At any time when this bacteria is exposed to the right environmental conditions, it could trigger the viral DNA taking over the cell and then us entering into the lytic cycle. It could happen at any time. Okay, and then we'd have, just following the lytic cycle. Environmental factors for bacteria could be something as simple as getting some radiation, whether it's ultraviolet radiation or something else that could weaken the cell, and the virus kind of sees this as its chance to take over, is a way that you could explain it. With this lysogenic cycle. Other times with the bacteria, it could just be a chemical change, so there may be some chemical in the environment that could be perceived as weakening the bacteria, and then the virus will take over. Okay, so that's the lysogenic cycle for these phages. And now we're going to get into some more specifics of what happens with animals, okay, viruses that are in animals instead of within bacteria. And you know what, I think I'm just going to make a whole new lesson for that, for what the viruses do in animals.